In 1903, Michael Tzvedt, a Russian botanist working on photosynthesis, published a paper describing a series of experiments he had conducted on chlorophylls. In his paper, he announced he had discovered a way to separate the various pigments that occur in the leaves of plants. Even before 1903, it was known that these pigments contained green chlorophylls and yellow xanthophylls and carotene. However, separating them was extremely difficult, and complete separations were seldom, if ever, achieved. As part of his experiments, Tzvedt constructed a simple apparatus consisting of a narrowly drawn tube with a filter paper cap over the tip. He packed the column firmly with powdered starch and then filtered his extract of the plant pigments through it. As the solution passed through the column, bands of the individual substances separated into layers. Tzvedt noticed that the separation could be improved by following the pigment solution with a wash of pure solvent. The resulting distinct color bands of the yellow carotene, then the yellow xanthophylls, and then the green chlorophylls, once separated, were easy to collect and purify. In the ensuing 3 years, Tzvedt continued to develop his new method of separating plant pigments, and in his papers of 1906 he called the process chromatography, a name derived from the Greek words for color and to write. These seemingly simple experiments set the stage for a revolution in the means of purifying and separating mixtures. Although he did not fully understand how the process worked, it is now known that Tzvedt's original separation was based on the relative equilibria of the plant pigments binding to the starch and dissolving in the solvent. When the mixture of plant pigments was added to the top of the column, all of the different components of the mixture were absorbed by the starch. Then, as solvent was added to the top of the column, the pigments were washed off or eluted. Equilibria were established between the pigments in the solvent and the pigments on the starch. The most soluble and least tightly absorbed pigment, in this case carotene, dissolved to a larger extent than the pigments that were not as soluble or more tightly held, such as the xanthophylls and the chlorophylls. The soluble pigment carotene moved down the column with the moving liquid. As it moved down the column, the carotene encountered fresh starch and was reabsorbed on the column. Equilibrium with the solvent was again established as more liquid passed down the column. The more soluble pigment kept moving down the column and was eventually eluted from the column. Therefore, the most loosely bound pigment, carotene, moved down the column first, slowly followed by a band of the next most loosely held pigment, and so on. The distribution of any solute between two phases is related to the relative equilibria of the substance in the individual phases. This ratio of the amount of solute in each phase is known as the distribution or partition coefficient. Because this distribution coefficient is a ratio, in successive extractions of a solution, the solute being distributed can eventually be quantitatively removed from one phase to another. All types of chromatography are based on these kinds of equilibria. By keeping one phase, such as the starch, fixed or stationary and allowing the other phase, called the mobile phase, to percolate through the fixed phase, continuous equilibria can be established along the length of the column. Small differences in the distribution coefficients of the components of the mixture can give an analytical separation of the species. For Tzvedt, the choice of solvent and stationary phases was largely a case of trial and error. In fact, he tested over 100 solid phases before concluding that starch gave the best separation. The choice of stationary and mobile phases is still somewhat arbitrary. However, with a better understanding of the equilibria involved and the factors governing them, a more informed judgment can be used in selecting the phases. For example, an organic chemist encountering a mixture of relatively non-polar compounds will frequently separate the species with a strong adsorbent, such as silica gel or alumina, using organic solvents of increasing polarity, such as pentane, ether, and alcohol for the mobile phase. On the other hand, a biochemist who more often deals with large molecules uses gel filtration chromatography to separate the components of the mixture.
Even though the choice of stationary and mobile phases depends on the species to be separated, the procedures that must be followed to gain a good separation on a chromatographic column are common. The basic piece of equipment used in chromatographic work is a tube with a sintered glass disc at its bottom. This tube can vary from a few millimeters in diameter and a few centimeters in length to one much larger. However, a burette of the right diameter and length can be used equally well, providing a small plug of glass wool or cotton is used in place of the disc. The glass wool should be gently pressed into place at the very bottom of the column with a glass rod. To prepare it for packing, the tube or burette is clamped securely in an upright vertical position. Great care must be taken in packing, since this is the most critical operation of the entire procedure. An irregularly packed column will not give a good separation, regardless of the care taken during the remainder of the process. A thin layer of clean white sand is poured on top of the glass wool. This helps to prevent the finely powdered stationary phase from leaking through the glass wool or clogging the disc. Care should be taken to ensure that the sand layer is level. Any sand adhering to the side of the tube can be washed off with a small amount of solvent. If more than one solvent will be used, the solvent with the least eluting ability is used as the rinse. A slurry, that is, a mixture of the solid phase or adsorbent to be used, and a solvent, can be used to pack the column. The stopcock is closed. The slurry is then poured slowly into the tube and allowed to settle. It is also possible to add the adsorbent to the column without the use of a slurry. When a slurry is not used, the stopcock on the column is closed and the column filled about half full with the solvent. The dry powdered adsorbent is added slowly while agitating the column. This will help to make sure the column is packed in a uniform manner. After all the adsorbent has been added, the column is agitated a bit more, and a few milliliters of solvent are allowed to drain from the column into a waste container. The column is vibrated slightly as this solid is settling to prevent air bubbles from becoming trapped within the solid. As stated before, the main concern is for a uniform packing of solid. Any irregularities that occur across the diameter of the column will prevent the sample mixture from flowing evenly down the tube, and this will lower the efficiency of the separation. After the slurry is settled, the stopcock can be opened and the liquid allowed to drain until it is level with the surface of the solid. Whether a slurry or dry adsorbent has been added, at no time during the separation should the solvent level ever drop below the level of the solid. If this were to occur, air would come in contact with the adsorbing solid, and as more solvent is introduced, this air would become trapped in the column as bubbles. A layer of sand is now added to the top of the solid stationary phase to prevent the top of the column from being disturbed when the sample and the solvent or solvents are added. The sample is dissolved in a minimum volume of solvent, and a dropper is used to place the solution directly on the sand. Opening the stopcock allows this sample to be adsorbed on the column. As soon as this is accomplished, the stopcock is closed. A few milliliters of solvent are added to the top of the column, and the stopcock is opened to allow the solvent to enter the column. This ensures that the sample is completely washed through the sand and adsorbed on the stationary phase. Additional solvent, that is the mobile phase, is added to the reservoir above the column. And as the stopcock is opened, solvent begins eluting or washing the sample through the column. The flow rate of the column should be adjusted to give the maximum separation of the components in the sample. Two opposing factors govern the choice of this flow rate. If the solvent passes through the column too quickly, there will not be sufficient time for equilibria to be established. But if the solvent moves too slowly, diffusion will begin to occur, and this will also decrease the separation of the components. For analytical columns, the optimum flow rate will vary between 4 to 50 drops per minute. Once the rate has been set, the sample begins to separate into distinct bands which are sometimes colored. As these bands of sample move down the column, the leading edge or front of the band should be horizontal, that is, perpendicular to the long axis of the column. If irregularities are present in either the adsorbing solid or in the packing of it, or if the column has large trapped air bubbles in it, channeling will occur. This causes part of the band front to advance ahead of the major portion of the band. It may not be possible to collect each band to the exclusion of the next one, and therefore these conditions will destroy any attempt at an analytical separation. As the solution drains from the column, it is collected in small fractions. These fractions, although mainly solvent, also contain the separated components of the mixture. They are usually collected in sequentially labeled test tubes or Erlenmeyer flasks. If the components appear as distinctly colored bands on the column as here, then the collection vessels should be changed just before and after each colored component is eluted, as this ensures that each component is collected individually. More commonly, however, the bands are indistinct or colorless. When this is the case, small fractions are collected successively and individually tested for the presence of eluted materials. Some of the components of the mixture may not be eluted with a single solvent. In this case, a more polar solvent is added to elute the strongly adsorbed compounds still on the column.
As we said earlier, Sweet's experiment set the stage for a revolution in the methods of chemical purification and analysis. Beginning with the liquid-solid or adsorption chromatography of Sweet, many types have since been developed. The study of chromatography now includes gas-solid chromatography, gas-liquid chromatography, liquid-liquid chromatography, ion exchange chromatography, paper chromatography, thin layer chromatography, and gel filtration chromatography. Michael Sweet put together his first column empirically without understanding why it worked. The theoretical basis for chromatography that now exists is largely a result of the pioneering work of Martin and Singh, for which they were awarded the Nobel Prize in 1952. The ability to separate and purify compounds by chromatographic methods has been fundamental to our present understanding of chemical properties and reactions both in the laboratory and in nature.